So today what we're going to discuss is how one can manipulate DNA sequences, but more importantly, under what circumstances one would manipulate DNA sequences of living organisms and how one would do it. Now, when it comes to crops or domesticated animals or anything like that, typically what one does is genetically modify an organism, and the way we talked about that is that could be done with a transposable element which can carry a payload into the genome. One can genetically modify, in this particular case, sheep, or one could particularly genetically modify a soybean plant, like we discussed in the last class, to mediate something called vertical transmission. So if you genetically modify this sheep, you would like to allow it to generate more sheep that have that mutation in it, or genetic modification. Or if it's a corn plant that has Roundup resistance, you would like that corn plant to establish a stock of corn plants that all are resistant to Roundup. So that property of transmitting genetic information from one generation to the next, which you would mediate through perhaps a transposable element, that is called vertical transmission. But there's another type of transmission of information, of genetic information, across organisms, and that is called horizontal transmission. And horizontal transmission basically means information moving from this individual to this individual to this individual. Now, horizontal transmission is often achieved through infectious agents, right? Where basically an infection moves from one individual to another, but there are ways in which DNA sequences can spread across individuals, not vertically but through horizontally. Now, what tool have we discussed that can enable genome hacking to modify the genetic code through vertical transmission? The genetic tool that we talked about were transposable elements. Because what you can do is you can genetically modify the germline, right? So there's sort of two aspects of what you can modify in an individual. You can modify the germline so that it would transmit it to the next generation. And that's how we do make genetically modified organisms. So we would modify a strain of cows or pigs or corn or soybean or so on and so forth. And the way we would do that is we would use a transposable element because they can carry payload into the germline, and then they can transmit that to the next generation. But obviously, while that might be considered okay for crops or different animals, it's not clear if that is a very ethical thing to do in the human population, to modify the germline. Because what that would mean is that if an individual modified their germline, they would transmit that modification to the next generation. So what might be better then is if one were to actually pursue genetic modification, one might rather not just simply modify the germline, but one might rather modify the entire soma. And by the soma, I mean the body. But how does one do that? So I'm going to leave you with this little meme from Morpheus. What if I told you we could modify the genome of an individual? How do we do that? How do we modify the genome of a single person without modifying their germline? And the way we do that is exploit something that already has the ability to modify the cells in an individual. And the things that have that ability are viruses. We've been talking in this class about genome hackers, basically enzymes or parasites or anything like that that can modify the DNA sequence of a target. Viruses are genome hackers because they can enter genomes, but they're not just genome hackers that just can transiently do that. They're infectious, meaning that they have the potential to modify the genetic material in potentially a large number of cells and potentially across multiple individuals. And viruses do a great job of this. These are all the different types of, not all the different types, but there's a many different types of viruses. Here's a coronavirus, SARS, HIV, measles, rabies, herpes, variola, Ebola, the thing's a monster, papillomavirus, dengue virus, hepatitis C, hepatitis B, hepatitis A, parvovirus, rotavirus, adenovirus, influenza, so many different kinds of viruses. All of these have the ability to potentially modify the genomes of who they infect. So the question is, if one were to think about trying to modify the genetic constitution of an individual using a virus, now that sounds dangerous, what virus would you use? So that's the topic of what we're going to discuss today. So in order to think about that, it's important to think about how we classify different types of viruses. There's sort of two schemes that I show here. On the top, I show a scheme which is centered on this idea that all viruses have to make mRNA. And the reason all viruses have to make mRNA is they have to make a nucleic acid that gets translated into proteins so that it can propagate itself. So the question then becomes, what is the source of mRNA? And we can define viruses based on that. You can have double-stranded DNA viruses that can infect the cell, and double-stranded DNA, when it gets transcribed, it makes mRNA. You could have single-stranded DNA viruses that when they infect the cell, they have to make double-stranded DNA, and then that double-stranded DNA gets transcribed to make mRNA. One could infect the cell with a double-stranded RNA molecule, and if those two strands are peeled apart, then the mRNA is exposed. One can actually express a single-stranded RNA molecule that infects the cell, generates a single-stranded RNA molecule of the opposite strand, and then that can then generate mRNA not through transcription, but through reverse transcription, right? Because you have RNA to RNA here instead of DNA. So here we have DNA, so normal transcription DNA. Here we have a minus strand of RNA that generates a plus strand of mRNA. You can infect directly with the minus strand of mRNA, or minus strand of RNA. You can infect with a plus strand of single-stranded RNA. DNA can be made from that through reverse transcription. Then DNA polymerase can copy the DNA, and you can make double-stranded DNA, and then you can make mRNA that way. Double-stranded DNA can make single-stranded RNA, which can then also be processed into mRNA. And this classification scheme is known as the Baltimore classification scheme. It defines the different ways in which mRNAs can be generated. All of these represent the infectious form, and these represent the form that exists within the cell that is replicating the virus. Excuse me. You can also break it down this way. You can also break down RNA viruses, reverse transcribing viruses, or DNA viruses. RNA viruses don't make DNA at all. That can be RNA that can make RNA, and in that life cycle they can make minus strand RNA, double-stranded RNA, or plus-strand RNA. Then there's reverse transcribing viruses. We talked a little bit about retrotransposon, similar type of thing. RNA can make DNA, and then the DNA can make RNA. All right, so this includes HIV and hepatitis. DNA viruses, obviously they make mRNA, but their replication cycle is DNA to DNA, and the infectious particles are comprised of DNA. Going back here, hepatitis C and SARS viruses, coronaviruses are of this kind. So this is a more detailed view of the life cycle of some types of viruses, double-stranded RNA viruses, plus-strand RNA viruses, or retroviruses. In this particular case, you can see that they're always localized in some sort of capsid. So there's a cargo in here. And this is important because when we talk about gene therapy, we're going to imagine that we're going to be able to put some sort of cargo inside this viral particle that would not be infectious, but instead would carry a gene into an organism. So in the case of double-stranded RNA viruses, This represents a double-stranded RNA. Inside a capsid, there's a receptor that recognizes it. It enters into the cell. The double-stranded RNA nucleic acids end up producing a bunch of plus-strand RNA that gets translated into all sorts of different proteins, and those proteins then package more copies of the virus to leave the cell. Plus-strand RNA viruses, right when they enter the cell, an RNA molecule is available for translation. It makes virion proteins as well as replication proteins. The replication proteins go from plus strand to minus strand to plus strand RNA again through RNA synthesis off of the RNA template, and then those get packaged then into these viral particles. Retrovirus is another viral particle that has an RNA molecule in here, enters the cell again through a receptor. The viral particle breaks up. The RNA molecule is exposed to reverse transcriptase. The DNA, and this is important, here the DNA actually inserts into the genome. Then transcription occurs. So the DNA is inside the genome. Transcription occurs. The RNA molecule generates through translation all of the particles and packages itself. Now, if one were to think about how one would use viruses to put DNA into an individual for gene therapy, which ones of these might be more important? The big distinction here is whether or not you integrate into the genome or whether you just simply express RNA, which might be your gene, or you insert DNA, which might be your gene. Okay, so those are some things to think about. So if you're going to do genetic therapy or gene therapy, these are I'm going to walk through all of the things that one must think about. So first off, the goal here is to use a virus to put genetic information into an individual. And the reason you use a virus is because of this. Viruses have this special ability to go into cells and dump nucleic acids into cells. If you hijack a virus and and obviously you don't want to put an infectious virus into somebody, but if you can basically hijack the virus so that instead of putting infectious material, it puts a gene into somebody, then that might be beneficial. But in order to do this, we need to think about how one would design a virus to inject genetic information into a target individual. What matters here is something called tropism. What cells can the virus deliver its contents to? All right, so for example, we all know that SARS-CoV-2 does a very good job of entering lung tissues. All right, so does influenza. And so that's a form of what is called tissue tropism, right? In which a virus has a special ability perhaps to infect a lung, but not, say, for example, a brain. All right, so influenza virus normally infects lung tissue, but not brain tissue. So that's a tissue tropism. So if one were trying to modify the genetic sequences of a brain, one might not use an influenza virus. Then there's cellular tropism. What type of cells can be infected? HIV, for example, normally can infect macrophages, but not neurons. And then in addition, there's host tropism, right? Because some viruses can infect some organisms. For example, the myxoma viruses can infect rabbits, but they can't infect humans. So you might not want to use a myxoma virus, for example, for genetic engineering of a human. A second factor that matters, and I briefly touched on it, is whether the gene that gets inserted is going to be integrated, meaning going into the genome, or whether it's going to be episomal. An integrated one will insert into the host cell genome in some location. So here you have a cell, the gene has been inserted. Now as this cell replicates, then all daughter cells have inherited that insertion. And so this would lead to a stable event in which the gene got integrated directly into the genome. But one can also put DNA molecules into the nucleus that don't get incorporated into the genome. And this is called non-integrating or episomal. And so as the cell divides, the circular DNA molecule, which might have your gene of interest, will slowly be diluted out because it doesn't necessarily replicate within the genome. So after many rounds of cell division, the transgene meaning the gene that you're inserting, basically sort of dilutes out from the cell population. So it's worth thinking about what the advantage of these different ones are. You might think on the top, oh, it just inserts a gene into the genome. That must be much better. But what's the cost of that? Well, the potential cost of that is you don't necessarily know where that gene is going. And that gene could insert into another gene, for example, that is controlling cell proliferation. And if you cause a mutation in a gene that regulates cell proliferation, what can you potentially cause? You can potentially cause cancer. So these are two things that one needs to think about, whether you want your gene integrated or not integrated into the genome. Another factor is the viral-like particle and the delivery mode that you're going to use for putting to mediate gene therapy, is it likely to elicit an immune response? Here we have a virus, recognizes a receptor, goes into the cell, produces what are called antigens or proteins. Now, it's possible that those proteins might get presented on the surface of the cell and be recognized as an antigen, and this could elicit an immune response. And perhaps even the cell that has taken up this viral particle may be targeted for destruction. If you elicit an immune response, two bad things could happen. One bad thing, which might not be so bad, is the cells that you inject your genetic payload into might get destroyed, and they might not be a very effective method. Another thing that could be harmful is that you could elicit a generalized immune response in which lots and lots and lots of cells are attacked and the immune system goes haywire. Okay, another thing that one wants to think about. I showed you at the very beginning different types of viruses that have all different sizes. All right, some are very large, some are very small. Well, the size of the virus, in fact, determines how much DNA sequence you can put into there. Adenoviruses, okay, shown right here, which are commonly used in research and in a little bit of actual gene therapy, you can put about 7.9 kb in there, or about 8,000 base pairs. Lentiviruses, same thing. You can put about 8,000 base pairs. AAV viral particles, they're tinier. Right? They're much smaller, and you can't put as much DNA sequence into it. You can only put about 5,000 base pairs. Another thing that one wants to consider is how are you going to get the gene therapy into the individual? There's direct delivery. So, for example, one can take a therapeutic transgene, you can pack it into a virus, and you could basically use that as a delivery vehicle and then inject that right into the patient. You have to make, millions and millions of these particles, but if you put your DNA sequence that you want in there, the virus will deliver that if it gets injected, perhaps into the liver. So you have to make sure that the virus has a way of basically targeting the tissues that are of interest. Another thing that one can do is do something called cell-based delivery, where you don't directly inject it into a human, but you could take an individual, take their stem cells out, grow up their personal stem cells, maybe from their liver, maybe from their pancreas or whatever, grow up their stem cells, and then deliver the gene through the virus into the stem cells, grow up the stem cells, and then re-administer them to the patient. So this is called either in vivo, which is direct, or cell-based, which is ex vivo. So, these are the things that one wants to consider when thinking about how you would do gene therapy. Another thing that you would want to consider is what is the nature of the way of what you're trying to fix? Are you trying to restore the function of a defective gene by simply expressing maybe a wild type or normal copy? Are you trying to knock down the function of a harmful gene? Suppose you have a tumor that has a gene that's overexpressed and is driving cancer formation. In that particular case, you might want to, instead of expressing a normal gene, you might want to knock down the function of the harmful gene. Do you want to enable resistance to infection? You could use gene therapy for vaccination, and this is something that's being investigated right now in the time of COVID. And then the other thing is one might simply want to actually just kill cancer cells. How could one use gene therapy for that? Another thing that needs to be considered is how you deliver what is called a replication defective viral payload. All right. So if you're going to administer gene therapy, you don't want to inject an infectious virus into somebody. What you want to do is you want to inject a virus that is in a capsid that doesn't have any ability to replicate. And so there's ways of doing that, which I won't get too much into the details of. But if one has something called a packaging vector and an envelope vector, the packaging vector and then a third generation transfer vector, you can basically inject this set of different expression vectors into a cell line, and these will all be expressed to produce the viral proteins. The viral proteins will generate a capsid, and then inside there you have the transfer vector, which has your RNA or DNA of interest in there. But altogether, this doesn't produce an infectious particle, because what it does is it basically generates viruses that have the DNA payload that you want, but it doesn't have any of the material that would make this viral particle infectious. And so basically you load up these infectious particles with your gene, and inside that gene, that DNA sequence, there's nothing infectious in it. Okay, so these won't have the ability to sort of make more and more and more copies of it, and so it delivers a payload in one dose to the cells that you're interested in. This is called a replication incompetent lentiviral particle in this particular case, but one can do it in different ways. So these are the things that one might want to consider. If you want to use an integrating approach, you could use a retrovirus or a lentivirus. It's often thought that these are less likely to induce a strong immune response, which could be harmful. In terms of their tropism, like what types of cells that they can go into, retroviruses usually can just go into dividing cells, although lentiviruses may be both. The problem with an integrating approach is that it has what is called potential oncogenicity. And oncogenicity, onco means cancer, so it has the potential ability to mediate cancer. But the advantage of it is that because it goes into the genome, it has long-term gene expression in dividing cells in the case of retroviruses, or long-term gene expression in most cells. Now, episomal approaches, these are non-integrating, can be mediated with AAV vectors. People have proposed herpes vectors or adenovirus vectors. AAV vectors have low immunogenicity potential, but the herpes viruses and the adenoviruses have high chance of inducing a strong immune response that could be harmful. They go into different cells. Herpes simplex viruses can enter neurons, which a lot of different viruses can't do. AAV vectors can go into dividing and non-dividing cells, as well as adenoviruses. But as I said before, the adenoviruses have small packaging capacity, all right? And there's a concern that the adenoviruses have this ability to induce a strong immune response. And so these are some other advantages. AAV vectors are thought to be non-inflammatory and non-pathogenic. The herpes viruses approaches have a large packaging capacity, so you can put a lot of genetic material in there, and the adenoviruses efficiently move into most cells. Now, I want to talk about a very famous example of gene therapy using viruses. There's a disease called ornithine transcarbamylase deficiency. And what this disease is it's an enzyme dysfunction that causes accumulation of ammonia. It's X-linked recessive, and usually it's lethal. But there was a young man named Jesse Gelsinger who was one of the first humans, one of the first people to receive a gene therapy. Other people had as well, but this was in the early stages of gene therapy. And he had an interesting situation. He had what is called somatic mosaicism for ornithine transcarbamylase deficiency. All right, so somatic mosaicism means not all cells have this defect. All right, so normally he wouldn't have lived to be as old as he was in this shown in this picture, and because not all of his cells had this defect. A large number did, but because of that, he was a good candidate for the first person to receive gene therapy to express this particular enzyme, right? So I said it's an enzyme dysfunction. So if you express the enzyme ornithine transcarbamylase, you could potentially cure this individual by allowing the ammonia to not be accumulated. So Jesse Gelsinger volunteered to help others overcome this disease, and he was treated with a disarmed adenovirus vector expressing the normal gene. And so here's the adenovirus vector, and a very large dose of this adenovirus vector, which expresses the normal gene, was injected into his liver. However, soon after that, he had a severe and overwhelming immune response. He had multiple organ system failure, and he died 4 days later. And this sent shockwaves through the gene therapy community. And it took a long time for people to actually continue investigating this. It basically set gene therapy work, it set it aside for almost a decade. The report that was done by the FDA to figure out what was the reason for why he died found that there were many cases of malpractice actually that were happened in this case, which is very sad. So first off, the Food and Drug Administration report found that prior to the injection of this gene therapy, he had very high levels of ammonia in his system, which should have in fact led to an exclusion from the trial. There was a failure to report previous symptoms of prior volunteers, and in addition, monkeys had received doses similar to him, and actually some of those monkeys died. And because of that, the person who ran that trial, James Wilson, had his practice of medicine revoked for 10 years. So this is a huge tragedy that set a lot of thoughts about gene therapy aside, and people thought for many years that gene therapy would never happen. But in the past few years, there's been a significant amount of success because people have been more clever and smart about how they administer gene therapy. So, there's a bunch of cases where people have actually had gene therapy administered into the eye to mediate improved vision. A few years ago, FDA approved a novel form of gene therapy to treat patients with this rare form of inherited vision loss. So this has been approved by the FDA. There have been cases where hemophilia has been cured with gene therapy, and a couple years ago in 2017, it was declared the year of gene therapy breakthroughs. And so there's a significant amount of movement back in the field of gene therapy, even though we had that tragic scenario with Jesse Gelsinger almost 2 decades ago. Now, one thing that I want to finish up with is that gene therapy needs to be sort of developed on a case-by-case basis for diseases that are very rare. All right. So what that means is for rare diseases that you employ gene therapy for, there's not going to be a lot of patients, right? And so to develop a technology for just a few number of patients, it can be very expensive. And so there are different syndromes and different gene therapy efforts that have been being pursued, and these are the names of different companies that are pursuing different gene therapy for different types of syndromes and diseases. One can see that the larger the number of patients, the expected cheaper the price, but it's still expensive. So For this, Gilead's the same company that makes remdesivir, actually. This company is developing a gene therapy for a particular disease. This thought to be maybe probably about 7,500 patients that will probably be eligible per year, but it's going to cost about, $300,000 for treatment. And this Novartis treatment, there's expecting maybe 300 patients per year will be eligible for that, and that's going to cost about $500,000. And you can keep going up, and the fewer patients that are available, this was the one that I talked about a moment ago for the blindness. Fewer than 30 patients a year are thought to be eligible for this, but it's going to cost probably about $900,000 for each therapy. And this particular one, probably fewer than 10 patients every year, and it's probably going to cost $1 million for each gene therapy treatment. So, the cost of these things is something to consider and be aware of when thinking about how one would administer gene therapy. Okay, well, that's a question that we'll ask in the next class, and so that is the end of this discussion about viruses and gene therapy.